Good evening, everybody, and welcome to the Prostate Forum of Orange County. We are proud to present tonight Dr. Mitchell Gross, who is the Senior Director of Translational Research, Ellison Institute for Transformative Medicine, and Associate Professor of Medicine at the USC Keck School of Medicine. He's going to talk to us about the genomic revolution and the use of genomes in diagnosing and prescribing care for a wide variety of diseases, but specifically prostate cancer. With that, the doctor has advanced degrees in medicine at Baylor College in Houston, Texas, and he has a Ph.D. in molecular biology from UCLA. He completed his residency in internal medicine at UCLA and a fellowship in hematology and medical oncology. We're going to have a busy night tonight, so I'm not going to take up more of your time other than to say here's how we run the meeting. Please mute your mic during the presentation and during the question and answer. I'm going to mute myself as well. We cut down on a lot of background noise that way. Our presenter will go on for 45 minutes to an hour. During and after that time, you are invited to post your questions and comments in chat, which I'll open up here in a minute. I will read those questions and comments, and the doctor will respond. If we have a need for clarification, we will ask you to unmute yourself and speak directly. This way, we have an organized question and answer session. Thank you very much for doctor for joining us. I'm going to mute myself. And we look forward to your presentation tonight.
Okay, so hopefully everyone can see my screen, and there's a picture of our institute there. It's called the Ellison Institute, which is in West Los Angeles. So it's close to the 10 freeway, but not in Orange County. And we're right on the Expo line. So that's the train there. But so we're in West Los Angeles. So what I thought I would talk about today is really with my background in molecular biology and as a treating physician for prostate cancer, how those two things come together. And this is really what we refer to as the genomic revolution of care for cancer, especially prostate cancer. So if we think about, a revolution, maybe comes to mind as like the Industrial Revolution, like in the upper right, where there was basically rows and rows of women or men, usually women, sewing clothes, but now maybe it's just rows of robots. And so the medical revolution, certainly the genomic revolution, is kind of exemplified by, we used to just maybe examine and look at patients with a flashlight, but now we have so much more information, and that's really the magic or the kind of the promise of genomic therapy. But we'll talk about this. The most important thing is not can we understand the genes, though that is important. The most important thing is, are they actionable? Can we find things in the genes that will give us a certain treatment for a certain patient? And that's really what I want to focus on here tonight, is the actionable genetic mutation. Okay, so the overview is we'll talk about genetics and genomics, especially related to cancer. We'll talk about actionable mutations, and we'll talk about specific therapies that will take advantage of this. And we'll talk also about the promise, but also the limitation. Okay, so this is to represent that we're kind of going back to school here a little bit. And what do we mean by genetics or genomics? So genomics is the study of all the genetic information in an individual. We know now that when we say genes or genomics, what we're really talking about is the DNA sequence, the information in the DNA that controls the basic functions of cells. And you can have mutations or changes in that DNA code that affects how a cell can grow, divide, move, or spread, or how a cell that makes up a whole organism can do those things as well. So for us as humans, there's basically 3 billion bases of DNA, 20,000 genes, On average, the cancer cell has about 4 mutations. So that's what we mean by genomics. And then this is sort of the revolution of genomics was really enabled by technology. So this is the kind of figure that represents that. The first time we sequenced a human genome, which was published around the year 2000, it cost $300 million to do just one example human. Nowadays, we can do it for about $1,000 or a little bit more, $1,500, and we can do any one individual. So in the last 20 years, been tremendous technology advances that really kind of drive this revolution, just like the Industrial Revolution was driven by industrial technology. So when we talk about the cost of doing this, or the cost of understanding a gene, it really depends on what we're hoping to sequence, or what we're hoping to how many pieces of DNA, how many bases of DNA. So if we were to just sequence one gene, on average about 10,000 bases, and the cost of the chemicals can be as low as $5. But this is a test that could be sold for potentially $1,000 or $100, for insurance and those things. If we were to do more genes, like what's called targeted genes that, for example, would be important for cancer, again, the reagents might only cost maybe $50, but the chemicals, that is, but the information and the time and the other things go into it. It might be $2,500 or more. And then for some situations with research, but also sometimes in patients, people do the whole genome. And as I said, that could be 3 billion base pairs. The cost could be less than $1,000 or less than a few $1,000s. But it could be sold, and there are some companies doing it for around $10,000. So again, huge technology, huge change in cost. Okay, so the other thing about going back to school here, as we talked about what genomics is, I want to talk about what is the difference between germline and somatic mutations. So when we talk about germline mutations, and we'll talk more about those are the mutations that are in all our cells in the body, meaning that it could be passed on to sperm or eggs. That's the germline. It could be passed on to the next generation. So germline means all the normal cells in the body. And also the germline, the important thing is this is shared by fathers to sons have 50% of the same genes, mothers to daughters, brothers and sisters about 25%. So these are the genes that are shared between families, and typically we talk about like inherited genetic risk and those kinds of genes. This is what we're referring to. This is germline genes that are in all the cells. The other thing to think about is what we call somatic mutations or somatic changes. So somatic changes are changes that are specific only to the tissue. So for example, this would be for if it was a prostate cancer that was removed from the body, it would be the sequencing of that cancer, looking at genomic changes in the cancer. These are things that are not in the normal tissue, but are only in this case, the cancer tissue. And these, of course, are not transmitted to the offspring. So a patient or someone has a mutation in the cancer that is specific to cancer, it's not shared by a brother or a son or a daughter. This is the specific in the mutation in the cancer. So germline, all the cells in the body, these are the inherited genes that go father, son, brother, sister, those things. And the somatic, those are specific only to the cancer, okay? All right. So we talked about genetics in cancer and genomic changes. Let's, we can, now this has been done, we can look at, for patients with advanced cancer, advanced prostate cancer, how many mutations, what kinds of mutations can you see? And So this is one of the largest recent studies to do that. So this is, they looked at, I believe, something like 1,000 biopsies or removal of prostate tissue from patients that had metastatic prostate cancer, and then they analyzed for genetic changes in that tissue. And here on the right is like a pie chart of the mutations that were found, and these are a bunch of genes, and we'll talk a bit more about what they are, but the biggest one here is called BRCA2, that's breast cancer associated gene 2, and of course that means it's also found in breast cancer, but these are changes in the BRCA gene in the cancer, but other mutations in ATM, a gene called CHEK2, BRCA2. So none of these are going to mean anything to most anyone, but the important thing about these is that these are mutations or changes that have to do with keeping the DNA, what are called DNA repair or genomic integrity. These are kind of proofreading for the DNA. That's what a lot of the mutations are. So again, we go back to this thing about germline mutations. Who should be tested? You may know that you can ask your doctor or a genetic counselor, and they can order these tests for, do you have a germline mutation risk? This this is what might be kind of conventionally called genetic testing. So for prostate cancer patients, the recommendation is for patients that have what are called high-risk prostate cancer at the time of diagnosis, or if it's metastatic, if it's spread outside the prostate. Those are the patients that should have germline testing. The other thing is this Ashkenazi Jewish ancestry, because we'll talk about this, but Ashkenazi Jewish ancestry, this is Eastern European ancestry, they do have an increased risk of some of these breast cancer genes that are known as BRCA1/2 mutations. The other reason to do genetic testing is if there's a positive family history. And so that means for us that it would be as a man with prostate cancer, and if they had a brother or a father, so another male relative that had cancer at a young age, especially prostate, but even maybe breast cancer as well. But the important thing is it's not useful for other patients. So the majority of patients with prostate cancer have what we would call low or intermediate risk disease, and they're older at the time of diagnosis, age over 65. Those patients probably not necessary to do the genetic testing. So it's especially in the higher risk patients that we do it. The other important thing about germline testing for prostate cancer patients is, at least at the time of diagnosis, it doesn't really inform therapy. It's not what we would call actionable in the classical sense. So we can do germline testing, and we recommend it. We do it in many of our patients, but it may not be actionable unless it's for more advanced prostate cancer. But the majority of prostate cancer patients, the sort of average risk patient, generally it's not done. Generally it's not actionable, not helpful, but it may help inform families and other things. So again, we want to focus on an actionable mutation. Sometimes germline or SNAC testing could be actionable, but that sometimes could be looking at the normal tissue, but sometimes the cancer itself, what's called somatic mutations. A big problem for prostate cancer patients is that often we don't have that tissue. We don't have a piece of tissue that we can look at to see if there's mutations. We can send for the analysis. So we often look in the blood, what could be called a liquid biopsy, or circulating tumor DNA. So we look for mutation in the blood, we look for it in the tissue, sometimes we look for it in the normal cells, which would be germline. That could be from a normal white blood cell, from a white blood cell test, like a typical blood test, sometimes even from like a swab of the cheek, or we can take the cells from the swab of the cheek and use that as well. But that's how we do germline testing. Okay. So in terms of these testings and somatic testing, I wanted to talk with just give you some names. So there's a number of companies that do this, some institutions, some big hospitals do this. These are some of the common commercial companies that do it. Foundation One, which came out of Harvard, can do somatic testing in a biopsy of prostate cancer or a surgical specimen. MSK Impact is a Memorial Sloan Kettering in New York that can also, is a common place that could be done. There's a large company called Caris, and they have a test called Molecular Intelligence. A company that we use a lot is called Tempus, and there's a test they call Tempus XT. But basically, these are what are called targeted genetic tests or genomic testing, where there's hundreds of genes that are mutated in all cancers, not just prostate, and we could ask that the pathology, the hospital, the test, the biopsy was done, and we ask them to send the tissue to one of these companies. The patients sometimes do have some copay, or at Medicare, usually it's not a copay, and they'll send it out to the company, and it takes about maybe 3 weeks later. We'll get the report back that will look for mutations in these groups of genes, hundreds of genes. Okay. All right. So if we look for somatic mutation testing in prostate cancer, one of the things is there are lots of different genes, a lot of different changes. So this is a very complicated figure, but just the big picture is that each column, so each vertical column represents a patient, and then each row represents a certain gene. And what you can see is there's dots where there's mutations in these genes. It's really all over this grid. There's some that are very common, like the androgen receptor has a lot of mutations, but mostly there's just like a smattering of mutations all over, which means that most patients have differences in the mutations. If it was all the same, it would be the same color left to right across. So the fact that it's all over, it just shows in these 100 patients, there's many, many different mutations. And this is what's known as the long genetic tail of prostate cancer. It's just there's lots of different cancer. But even more to our point here is only about 20 or 25% of these mutations are what we consider actionable, have a certain treatment, and we'll talk about that. Okay. So this thing about actionable, I keep hitting on, it really means these are changes, genetic changes that have a certain treatment. And really there are two main kind of classes of these genetic treatments nowadays. One is called BRCA2 is the most common one, PARP inhibitors. A few others are related there, and then something called MSI high. And so these are in that sort of 20-25% of prostate cancer patients that have an actionable mutation. These are the two most common, what are called BRCA mutations, and below that would be this MSI high alteration. So what do we mean by that? So this is the BRCA pathway. This is the PARP pathway. This is why we think it's actionable. So what does this really mean? So here in the upper left, the panel A, this is this would be your DNA, that double helix, the sort of twisty place with the bases there. And let's say it's damaged because of sun or the radiation or just bad luck. Let's say there's a defect in that DNA, and that's what can happen. So the normal situation is the body has these proofreading enzymes that are really looking for this change. And what would happen is this big green blob that looks like a milk bottle would find these changes. That's called the PARP enzyme. And what that does is it recruits these other enzymes that are basically their job is to fix the DNA. So it finds any changes and will fix it. And then like the fixed DNA can go on and function. So this is a beautiful system that controls the DNA repair pathway. Okay? But what happens in cancer is that you can have a mutation in the genes that are part of this process, and the most common one is, or these genes are called BRCA2 or BRCA1. These are related to the DNA repair pathway. And so in cancer, if you lose that you have a mutation in that gene, what would happen is that either the DNA would try to replicate, and it couldn't. It would be basically blocked, or the cell will replicate and make more mutations. So you really need the DNA repair pathway. If you don't have it, either the cells will die, a normal cell will die because it sees this as a problem and it will die, but a cancer cell could replicate and could basically make more mutations, and this is partly of the problem in cancer. One mutation begets more and more. But if you have a PARP inhibitor, if you have a drug that inhibits this repair pathway, this bad repair pathway, then you have what's called a synthetic lethality. So I'm sorry, we can talk about this more, but so if you have a bad DNA repair enzyme like BRCA2 mutation, and then you also inhibit the other DNA repair by giving what's called a PARP inhibitor, then you have these two hits that prevent any really DNA repair, any DNA replication. And without those pathways, the cell will die. Basically, you'll have so much break in the DNA that the cells won't replicate. And so that's this issue of DNA repair and PARP inhibitor. If you have a defect in the DNA repair pathway, and you also give an inhibitor of that pathway, which is a PARP inhibitor, you have what we call two hits. And those two hits, or synthetic lethality, will kill cells that have the mutation, and only the mutation, and those are the cancer cells. So that's basically, it's a little confusing, but that's how the DNA repair system works. So fortunately, this has been, basically this idea has been exploited to take care of cancer for, maybe about 10 years. And specifically for prostate cancer, in the last 3 or 4 years, there have been a huge amount of research on patients that have DNA damage repair prostate cancer, and that we can treat with a specific drug. And so, I have to keep updating this list, and you can see it gets updated every couple months, at least lately. So the most common drug that's a PARP inhibitor we use in prostate cancer is one called olaparib or Lynparza. And this is the FDA approved indication, which is, I know, very long and hard to read, but basically it's advanced prostate cancer that has grown despite androgen deprivation therapy, and that has a certain mutation profile. And these are the genes that are known, the most common one, again, being BRCA2 mutation, either in the germline, but most commonly in the cancer. Somatic mutation testing shows a deficiency or an alteration of BRCA2. This was approved for prostate cancer in May of 2020, and then was basically approved again in a different situation to use with these drugs, with abiraterone, just this year, earlier this year. The other drug, rucaparib, that was the first, or about the same time, also for metastatic prostate cancer that is growing despite androgen deprivation therapy, and that has alteration in one of these two genes, BRCA1/2. More recently, a drug called talazoparib has been approved. There's a longer list of mutations, but similar: hormone refractory prostate cancer, castrate-resistant prostate cancer, that grows on a drug like androgen deprivation therapy or Lupron, and it's used with enzalutamide. That would be the approved indication that was just in June of this year. And very recently, just last month, this other drug called niraparib, again, another PARP inhibitor, was approved, similar castrate-resistant prostate cancer with certain alterations. So this is the current list of drugs that are approved that are PARP inhibitors for the actual mutation of a BRCA1, 2, or it could also be called homologous repair deficient prostate cancer. Okay, that's all well and good, but I thought we'd kind of give you a more specific example of how this could work. So myself with Mary Duong, who was my physician's assistant, who helped me set up this meeting. This is an example of one of the patients we treated a few years ago. So this is a patient with prostate cancer that had surgery and radiation, but the cancer came back after that. And this is here in the picture. This is what's called an oxygen PET scan, and this bright spot in the middle is a mass that is in the prostate area that had regrown after prostate cancer treatment. So we didn't know what this was, so we had to biopsy it, and it showed to be prostate cancer. And partly why we did that is his PSA was going up here. So the PSA was 1.4, but he had a prostatectomy. His PSA should be basically 0. So that was the concern that told us we really should do something and do a scan. The scan showed that mass that proved to be prostate cancer in the area of the prostate, what's called local recurrence. But we did send it for somatic testing, and it did show this BRCA2 loss. So this is, again, if there is a mutation, this is the most common one, BRCA2 loss, and that was back in 2020. Then we treated the patient with a PARP inhibitor. In this case, I think it was olaparib. But what happened is his PSA came down, and even better, that mass completely went away. So here on the right is after a few months of treatment, about 5 months of treatment, the PSA came down from 2 to about 1. But more than that, this mass completely went away. So it was really a very pronounced response to a PARP inhibitor. So if you can identify the mutation in the right patient, it can have really tremendous effects. Okay. So this is the specific example of actual mutations. Often they're related to DNA repair, PARP inhibitor, and homologous repair deficiency, BRCA2. Most common is one. Let's talk about the second example here, which is the immune checkpoint inhibitor. Okay. So what does immune checkpoints have to do with prostate cancer treatment? So there's something called the mismatch repair deficiency pathway. So similarly, this is having to do with the proofreading of the DNA, but a different system, a different kind of mutation. But the body, the cells, have this ability to see if the DNA, which is supposed to be a perfectly smooth double helix, has any kinks that could represent like a problem where there's even just one base, like one little kink in the DNA, where instead of, let's say, an A, it's a G, or instead of a C, it's a G. So like it can make these kind of small changes in the physical structure of the DNA, and it can kind of find that. And if it does, it knows how to fix it. What it does is basically it cuts out the bad piece and puts in a new piece, and does it in a way that keeps the sequence correct. So this is what's called mismatch repair system. But if you have a defect in that, again, you can develop mutations that I think that's represented here. So if you find these mutations in the DNA just from the environment or from what's bad luck as the chemicals replicate, but you need the ability to fix it. But if you don't fix it, the cells will make that mutation and carry it to the daughter cells, and the cells will keep having more and more mutations. So the importance of the mutations is the mutations allow the immune system to see cancer, if you will. That if you have cells that have a lot of mutations, your immune system is basically programmed to get rid of those mutations and treat those cells. And so that is kind of a good thing for the immune recognition. But so this would be one complicated way to represent it down here, but I thought of an easier way to represent it, and that's in this little cartoon here. So here's your immune system. The nice little blue guy is the good guy here, and he has to find the cancer. And this is a big problem in prostate cancer, because remember, the prostate cancer is you. It is the self. It's not doing the right thing, but it is, from the immune system standpoint, the same as you. And you don't want your immune system to attack you all the time. That causes disease. But if it was a cancer, you want to be able to find it. So the microsatellite instability, the mutations, give basically the immune cell the wanted poster. It can find the bad cells in all the billions and billions of the good cells in the body. And so the immune system's job is to go out into the body to find these bad cells that have the mutation, and that's again what microsatellite instability gives the cancer cells mutations, and gives the immune system the ability to find it. But what happens is, so here's in the middle panel here, the immune system has now used the wanted poster, found the cancer cell, but the cancer cell has basically a defense. And that's this baseball bat, which is called a checkpoint inhibitor, and that will check the immune cell from trying to attack it. So that would be bad in the normal situation. But here we've given the cancer cell a protection. In this case, it's an antibody that prevents prevents the immune inhibition. And that's called a PD-1 inhibitor, or a checkpoint inhibitor. So the checkpoint inhibitor is basically the protection from the cancer cells from inhibiting the immune response. And so what happens here in the lower right, the immune system has found the mutations, found the cancer, and is protected from the immune. It's called the checkpoint, where the bad cells are trying to prevent cancer cells destroying them, and then the cancer cells the immune cell destroys the cancer. So this is kind of the idea of why hypermutation that is associated with microsatellite instability is basically the immune targeting mechanism as well, sort of like an Achilles heel if the cancer cells do have it. Okay, so this is kind of a complicated but maybe a very enticing idea to basically, promote or train the immune system to attack cancer. And in fact, this is a Nobel Prize winning idea. So basically Jim Allison, who was at Berkeley and then also at MD Anderson, spent his lifetime, championing this cause, and led to a whole slew of drugs that are what are called checkpoint inhibitors, along with his Japanese Dr. Honjo, and they won the Nobel Prize for this back in 2017. Okay, so there are a list of these drugs that are used for microsatellite unstable cancers, and the important thing there is the term microsatellite instability, or MSI high cancers. I don't say prostate cancer. I just say any cancer that has this property, this genetic change, MSI high. So this is a really important but very still small concept in cancer treatment that are called histology agnostic. So in this case, we don't care where the cancer started. We don't really even care where it is. But if it has this genetic change that we can identify, we know it can be treated with one of these drugs. So this is a small list of drugs, but there are drugs that are doing this, and these are basically 6 drugs for 4 different mutations that have been approved that are tissue agnostic. Again, it doesn't matter where the cancer comes from. So for MSI high prostate cancer, that probably only represents about 1 or 2% of metastatic prostate cancer, so it's very unusual. But if we do find it, we can treat those patients often with a checkpoint inhibitor, pembrolizumab or dostarlimab, which are the 2 there. And then even rarer, there are certain specific mutations in prostate and other cancers called NTRK mutations or BRAF mutations. And for those cases, there's specific drugs that can be used. So again, this is a really interesting development of genomically informed therapy, but still really a minority of patients that it applies to. And if we go back to this complicated figure where each column represents a patient, somatic mutation in a patient, and each row represents what that mutation is, you can see this huge smattering, but kind of somewhat fortunately, there are some groupings. And here at the bottom in the yellow or blue color, those are the patients that have a DNA damage repair or MSI high mutation phenotype or genotype. So maybe it's about 20-25% of patients overall, but that's some. And then there are these other groups of mutations that are common in prostate cancer and other cancers, and we're developing drugs against them. So like there's this column here, this row that's like purple, that would be especially what are called upregulation of a BET inhibitor, or a BET pathway would be active, and maybe there's a drug called BET inhibitors, a class of drugs. So we have one of those trials at our center, but others do as well. And then some of these other pathways. So again, there's promise, but there's some limitations. It's hard to keep track of all these mutations. So fortunately, there's all sorts of databases and things. So this is a relatively easy one to use called OncoKB. It's out of Sloan Kettering, New York. And you could put in a mutation and it could tell you if it's targetable and what the drugs would be. So again, this also gets updated regularly. And in the last 2 years, I've seen, probably another dozen drugs added, but this would be the current list. So last, I wanted to talk about PSMA as another target of therapy. So PSMA is a prostate-specific membrane antigen, so this isn't really a genomically informed therapy, but it turns out common in prostate cancer, this particular protein is expressed, and we can use this as a treatment, as a target for treatment. And one of the biggest new approvals in prostate cancer is something called Pluvicto, which is a radioligand pharmaceutical that basically has two sides to the drug. One side finds the PSMA protein expressed in prostate cancer cells, and then the other side delivers a radiation treatment called lutetium-177 that can kill the prostate cancer cells. So this is like another way to get around this problem of finding the cells, and this is a very interesting and new approach. There are other trials, but again, these are for advanced prostate cancer. There's a trial that we have at our center looking especially at certain mutations in the prostate cancer using an approach called AR-PROTAC from a company called Venus. So that would be one, that's the top one. We talked about BET inhibitors, again, very specific patient populations, but there are drugs being developed, and we have one at our center called NUV-868. There are other trials at other centers for sure, like the idea of an antibody that promotes the immune system, not passively, but actively, something called a BiTE, a bispecific T-cell engager antibody. There's some trials, AMG 160 would be one of those. So there's all sorts of ways that we're kind of using this genetics for understand the cancer, but also especially to hope for treatment. Yeah, and that's kind of the big summary. So every cancer has some genetic fingerprint, and we hope that we can use it to specifically target the cancers. We have a list of drugs. It's growing, but still not complete, that some patients we can have a specific drug for that cancer. The other thing which we didn't talk about, but typically these genetically informed therapies like the PARP inhibitors and like the checkpoint inhibitors have very low toxicity. So that's a fortunate thing about that approach. But there's still challenges. As I mentioned, only about 25% of patients will have one of these actual mutations. And also the list of drugs that we're choosing from is relatively short. So I wish we had more drugs, and we're always working to expand on that list. But the other things, we hope these drugs don't have toxicities or have very low chance of those toxicities, but they are toxicities for some of them. So this is the kind of thing that we are worrying about. So that's really the summary. We talked about the kind of the genetic revelation of prostate cancer, the revolution. I really wanted to focus on this thing about germline testing for patients that are high risk, and that's high risk or metastatic cancer, but not all patients. And then it's really important if patients do have a metastatic cancer or one of these high risk cancers, that we also look for somatic changes in the cancers or in the blood associated with them to see if we can find these actionable mutations. Because as good as these drugs are, we can't use them unless we know or we can identify patients that have that change, that mutation. Okay, that's it. So thank you, everyone, for your attention. I hope you were able to hear me okay. Let's see. So that's it. So any questions from the audience?
Neil, can I ask a question?
Yes, go right ahead, Doctor.
Yeah, thank you very much for a very, very nice talk. Helps clarify some things. Can you attach the PARP inhibitor to a theranostic agent?
Okay. So yeah, the question is, could we use the PARP inhibitor and attach it to some, there's a lot of these ideas of these, we call them bifunctional drugs, right? Like one side finds something, does something, the other side does something else. So a theranostic would be like, could you have a PARP inhibitor that somehow could identify cells that have this kind of mutation deficiency? It's an interesting idea. I've never seen it done or even researched. I think the problem is the PARP inhibitor is inside the cell, that it's a problem in the DNA right in the middle of the cell. It's very hard to probably image that, but I wouldn't be surprised if someone was doing it.
It would be nice to be able to get that PARP inhibitor to the targeted cell and not to the rest of the body. So
so
like
one of the advantages, and I'm sorry, I don't think I explained it so good, but that generally the cells with a mutation in BRCA, let's say BRCA2, are only the cancer cells. So that's one of the really advantages of the PARP inhibitor strategy, is that the normal cells have lots of backup pathways and they're not affected by the PARP inhibitors, but only cells that have a mutation in the let's say BRCA2 gene are affected. So it already is kind of programmed to only affect the cancer cells, more or less, yeah.
And the other question I have is, we're seeing intensification of treatment and everything else, it seems like. How's that working out with the PARP inhibitors or any of the other checkpoint inhibitors?
Yeah.
Or things?
Yeah. So that's a very good question. So if we were to look at kind of definitely the history of cancer, definitely the history of prostate cancer, what we would say is there's this like move to make the treatments further, move them up line, we would say. So for example, drugs like abiraterone and enzalutamide used to only be used after chemotherapy. That was the original approval. But now we use them in men that never had chemotherapy. And now we use them in men that are at the very beginning of treatment and hopefully will never need chemotherapy. So the same is true for PARP inhibitors. unfortunately, the current trials where we are right now, and this is the delay of stuff, are only for metastatic castrate-resistant prostate cancer, so only for the much more advanced. It makes perfect sense to probably try those earlier in treatment, and maybe even to not use androgen deprivation therapy or use them only with androgen deprivation therapy. And there are trials doing that, basically moving them up in therapy, where basically, let's say, hopefully patient maybe before radiation or with radiation, and that could prevent spread and those kinds of things. But none of those are ready. None of those are close to being ready. So yeah, there's efforts to move them up in therapy and combine them with sometimes radiation, sometimes chemotherapy, or sometimes these hormone treatments. But nothing is close. I mean, I think it's still a couple years away before those report out.
By the way, I apologize. I didn't introduce myself. I'm Dr. Metzger. I'm the pre-Skinner era at USC. So I know where you eat lunch.
Yeah. No, I've heard of you, I think, but because I've been working for 20 some years, but that's
Thank you
great
very
to see
much
you.
for that talk. That was great.
Dr. Gross, Dr. Gross, I have a question. If you had genetic screening and with no germline mutations, during in recurrence at a low level, you mentioned somatic genetic testing to see if there's anything actionable in the cancer cells. Is that recommended then? Would you do some kind of maybe a liquid biopsy and see if there's something, and do a genetic screening to see if there's something targetable there?
Yeah, so you definitely can. I mean, like all of these, it's a kind of qualified answer here, but for patients, if the cancer comes back after surgery or radiation, and certainly if it's metastatic, if it spreads to an area, a bone or something after, it is good to look for the circulating tumor DNA liquid biopsy. What the other part of that, though, for patients, if there's no metastatic site, so a common situation would be what we call biochemical recurrence. Maybe the PSA is 2 or it was 1, and after radiation or something, then it's not as likely to be informative. I mean, you can still do it, but it's not as likely to be positive. So the only real downside, luckily, it's just a blood test, so that's easy, but it's getting it approved for insurance and paying for it and those things. But yeah, it can be useful sometimes in those situations, the liquid biopsy.
Okay, thank you.
Right. Okay. Any other questions?
I think that this is all new territory for our very educated audience, and you may have left them speechless. And that's the sense that I have right now. Our folks are usually loaded with questions, but I think they're processing right now. And a new member of our group said, yes, he did leave us speechless. I think what we'll do is make a final call for questions. And if no one else wants to unmute and ask the question directly, we'll move on to some of the things that we do. I see Ira has.
I have just a quick question. Do you see patients in clinical practice?
Yeah. So I'm very fortunate to be a medical oncologist for 20 plus years and see patients as well as do research. So yes, we have a clinic called the Ellison Institute in West Los Angeles, and I'm very fortunate to have my right hand, Mary Duong, physician's assistant, who definitely helps me greatly with all of this. All the important stuff like making sure these genetic tests are covered with insurance, and minimizes the out-of-pocket, which is we didn't talk about, but usually there's definitely a cap at the out-of-pocket. for U.S. patients and definitely Medicare patients. Mary could even comment on that, maybe.
Another question. Are there any clinical studies that you're excited about now that you're working on?
Yeah, so specifically at our institute, there's a couple trials that we have open. I mentioned kind of briefly, and I didn't get too much into it. One is with our colleagues at St. John's and this company called Venus, a drug that looks for, that's especially good for patients with a mutation in the androgen receptor. So this is an early trial, so it's not proven, but the idea of the drug is that for patients that are advanced prostate cancer that have a mutation in the androgen receptor, that's really a common problem in these patients, that we can give a very specific drug. So it's kind of like a super duper Casodex, almost in a way, which has pretty low toxicity, and in theory should work against that cancer cell. So, this is something we test. It's, that's why it's a trial. And then we have a drug that is what's called a BRD2 specific inhibitor. So that's another drug target in prostate cancer. These trials are for like what are called phase 1 trials. So these are advanced cancer patients. We definitely treat other patients, but it's only really appropriate for patients where the standard treatments maybe aren't working. So those are 2 trials that we have open specifically relevant to this group. Let's see. I mean, one question which no one asked, but I think is very important as a clinician, is the cost of all this. So since we have really my expert here, I wonder if I could ask her a question about out-of-pocket costs for these genetic tests.
So for most of these genetic tests, the out-of-pocket is actually almost nothing. Most of them have very generous financial assistance programs where if you have non-Medicare, a commercial payer, depending on your income, they would just accept insurance as payment in full. You do have to fill out a form. Some of them, like Myriad, would, like I think up to $295 or $395 if you sign the form beforehand. Make sure you sign beforehand when the doctor's drawing it. Then you would be maybe liable for up to like $395, and anything above that they would talk to you. But they're very generous. They'll work with you to offset any out-of-pocket costs. Medicare, for the most part, will cover, as long as indicated, 100% of the costs. And as you know, Medicare is Medicare B and then Medicare supplement. So obviously, if you don't have a supplement insurance, you may be subjected to 20%. But almost everyone has a supplement insurance.
Yeah. So yeah, that is very important. Treating any patient, we certainly pay attention to potential cost as well as toxicity, but financial toxicity is a very real thing. So, that's why we don't order them all the time. We over order them, basically abuse the privilege. So.
Hi, I was diagnosed with ATM mutation. Any progress on that particular?
Yeah, so ATM mutation is one of those list of mutations that we think has to do with the DNA repair pathway. But what's been found in a lot of the trials so far is that doesn't seem to be as sensitive to the PARP inhibitors. It doesn't mean they don't work, but they're not as sensitive. So some of the particular PARP inhibitors, and there was this complicated table of all the drugs, some of them are only used, only a few of them are allowed, technically approved, if there's ATM mutated prostate cancer. But there have been a trial, we did a trial earlier this year that tried to identify ATM mutant prostate cancer with a specific drug. This particular agent didn't seem to work, but there's other agents that are being looked at. So yeah, there's certainly research in that regard, but right now, ATM mutations, there's only a few drugs used officially.
Thank you.
Right.
Anybody else? Final call? Doctor, I want to thank you for your time and for an outstanding presentation that brings hope to a lot of our people. We hope we can continue this for many more years. We think this is going to get more and more interesting as we go along.
There's stuff on the chat.
There's stuff in chat that just came in. Yes. Why don't we go to chat? Can you see it? Do you want to read it?
Yeah. So I mean, I know one question has to do with getting approval for this NGS testing. This is what would be called next generation sequence or somatic testing for metastatic prostate cancer, and they're having trouble getting it approved. I don't know, it's totally medically necessary. I can't see why it wasn't approved. I don't know what to say about that other than it should be approved. I mean, it's in the guidelines. There's what's called consensus guidelines that sort of judges what insurance cover should cover, and it is in the guidelines, most definitely. So I don't, it's hard to know what the paperwork said or what it was, but it should be approved. And that's like a slam dunk thing, I would think. And then this other one, which is very specific to a patient, someone with a PSA of 600 and a lung biopsy. Yeah, I mean, that sounds like definitely needs a medical oncologist, someone who's really a specialist in prostate cancer to take care of. I can't really comment otherwise, but yeah, very important to get a biopsy, to send that biopsy for somatic testing and to start treatment right away.
I want to urge everyone, particularly you folks that have not participated directly in this presentation, reviewing this at home, so to speak, to visit our website at Prostate Forum OC at prostateforum.org. Prostateforum.org is our website. Email us at prostateforumoc@gmail.com if you have questions, comments, want to know what's going on with our program, want to sign up for emails. This is particularly important for you that will be viewing this weeks, perhaps months into the future. Thank you very much, Dr. Gross. We look forward to having you back.
Thank you so much. Thank you for the questions. I really enjoyed it. These are all really good. I thank you for your attention. I hope I didn't overburden you, but it's something to think about and ask your doctors about definitely as well. So thank you.
I think it's very exciting. I think we're going to have many more meetings like this, and it's going to get more and more exciting as we go along. Thank you all very much.
Thank you and good night.
Good night.